Subject is very vast to understand the topic. Please make sure you listen till the end of the video, then you may understand. Hello everyone, I am your MM. Today I'm telling the topic as enzymes as double agents, the new mechanism discovered in protein modification. What is that one? Let us enter into the topic. Proteins are the workers in a cell and as the basic element of life are responsible for the most widely varying metabolic processes. In plants, for example, they take on an important function in photosynthesis. In order to be able to work purposefully, proteins change their chemical form after they have been produced in a cell. For example, through protein acetylation, when an acetyl group is transferred to the protein, the role of the driver is played by enzymes, so-called acetyl transferases. Not much is known, however, about the precise mechanisms underlying the process. An interdisciplinary team of researchers from Germany, Finland and France have now found 8 new acetyl transferases in a cell organelle in plants. In doing so, the researchers made a surprising discovery. The enzymes identified are catalytically active in the cell in a twofold way. In other words, they call as they facilitate reactions in different ways. It had previously been assumed that different acetylations were driven by different enzymes. They said this study shows an entirely new complexity within the enzyme machinery. I told the topic title also, the enzymes have double activities. Means, what is that? Means the double agents. The new mechanism they discovered in the protein modification. Okay, within the enzyme machinery, the new complexity they observed. Who says? Means this one, the Professor Iris Finkmeyer from the University of Münster, Germany, who led the study together with Dr. Carmelo Ziglione from the University of Paris, Saclay, France. The results strongly suggest that there are also other enzymes which can display such double activities in eukaryotic cells. For one of the newly identified enzymes, the researchers have already been able to demonstrate that it has an important function in photosynthesis. The long-term aim is to understand the process triggered by acetylation and perhaps make photosynthesis more efficient, says PhD student Annika Brunji from Münster University, who is one of the 2 lead authors of the publication, along with Dr. Willy Bienvenut from the University of Paris, Saclay. The study has been published in the General Molecular Systems Biology. Please, you can follow the Molecular System Biology General also. Okay, for the experiments, the researchers used the model plant Thelcrage Arabidopsis thaliana. They took a closer look at 2 important ways of protein modification, so called N-terminal acetylation and lysine acetylation. In these modifications, an acetyl group is transferred either to the end of the protein called the N-terminus or to the lysine residue. Both processes are very often found in proteins in chloroplasts, which are the cell organelles in which photosynthesis takes place. In order to find out which enzymes play a role in this process, the researchers first used a protein database to identify possible acetyl transferases, and in addition they located the occurrence of the proteins in the cell subcellular localization. In this way, they discovered in the chloroplast a new family of certain acetyl transferases known as G0. Yes, So much of this using quantitative mass spectrometry method, the researchers were able to show the enzymes are active in both N-terminal acetylation and the lysine acetylation, changing the proteins accordingly. Conversely, the researchers noticed that the reactive one of the enzymes through genetic modifications, neither of the acetylations function properly anymore. In further studies, the researchers aim initially to discover which functions the individually identified enzymes have. They expect to discover even more new processes in the coming years which these enzymes regulate in the chloroplast, says Carmela Giglione from the University of Paris, Saclay. Thank you. Please subscribe my channel and click like button, share with your friends to subscribe and increase. Please give an encouragement with your valuable comments. Thank you.